Here, actually, the conference theme is for influenza, but sometimes actually we need to. So yesterday we have heard wonderful talks, and also early this morning wonderful talk about the influenza efficacy of the new methods and also different the vaccine design strategies. Sometimes we need to jump out of this black box, this kind of influenza, and see if we can learn anything lesson or the successful philosophy we found from other viruses. That's why I'm here for actually bring the experience and also the result actually accumulating in my lab in the past couple of years about the HIV and how we use this HIV experience to infer actually improved the vaccine design for influenza. So HIV and the influenza, both are very important. The RNA viruses and of great importance of public health, that's for sure, and also the both actually the very the heavily glycolated. This I'm going to talk about more later on. But first, for you, because most of you are influenza people, so just to briefly let you know. After 30 years, skilled effort of the HIV, but so far we still haven't have any vaccine yet. So if you're interested towards the end of my talk, so I'll briefly talk about the current vaccine strategies and in the HIV field, and also why the strategies we tried before actually actually didn't work. So HIV, where I'm going to talk about is about the main group, which is the M group, and actually it's classified by the phylogenetic analysis majorly into 9 subtypes, which has A, B, C, D shows here based on the envelope, actually the protein, and also increasingly currently we see expanding of the HIV diversity imposed by the recombinant, and so that HIV vaccines actually designed currently becomes more and more challenging because we're not only dealing with some certain classification or subtypes, and which is already is very complicated, but also we to address the uncertain degree of the complexity imposed by recombination. So I will be focusing on 2 mechanisms that driving the extraordinary diversity of HIV. One of the glycosylations, the protein changes, which is also useful, and we could learn the lesson from HIV to infer the influenza vaccine design, and also about the recombination similar to the influenza reassortment. So first is glycosylation side, you know already is a very common form of protein processing, okay, like this form. And and very frequently we shorten its name into called a sequon. So if I later slides I will say sequon. Actually, I mean the unlinked glycosylation site. There's also like O-linked glycosylation site, but actually this one is better defined. So this is why we are focusing on first. So although this pattern of glycosylation site is very simple, asparagine followed by any amino acid followed by the serine or threonine, this kind of very simple pattern actually it could profoundly affect the protein expression, the structure, and also the function. So GP120, which is part of the envelope together with GP41 in HIV, that's the heavily glycosylated. So in this picture, you're seeing the black dots. So this is all the glycoproteins protruding from the viral particle surface. So HIV, that is the most actually heavily glycosylated protein natures. We actually at the first we think that's great. If we will solve this mystery in HIV, we can easily migrate the same analysis system to the influenza. This is what we thought, but actually turns out it's not that simple. I'll give you just one overview about the functions discovered so far about the glycosylation site, the sequon, which is first is to provide a self sugar shield, and secondly, actually the changes of the glycosylation site in terms of location and frequency, it can profoundly affect the epitopes. For example, so So the gain, if you're having additional glycosylation site, okay, so now you could probably ask me what's the reference, right, when you say gain or loss. So I will tell you that later on. So if you have additional glycosylation site in HIV, actually that's like can sterically mask epitopes. It's like the umbrella. In a rainy day, yesterday, like a rainy day, you hold an umbrella. So you have additional umbrella, so you can hide the epitopes that's sitting underneath of the umbrella. And also actual loss. Loss of the glycosylation site, you think, okay, great, now the umbrella is gone, so I can expose my epitopes so that immune response could be more efficient. This is part of the story. The other part is that actually the loss of additional of the glycosylation site that could lead to the tight packing of the glycoprotein regions, so that could reduce accessibility and facilitate immune escape. For example, in GP41, which is a very important part of this envelope in HIV, actually the epitope changes in the GP41 can actually dramatically change the glycosylation site. Okay, changes basically reducing the number, and so that actually could reduce the immune recognition. And so, and later on, I will talk about more how the viruses, HIV especially, use this mechanism, very simple pattern, right? And it's like asparagine followed by any protein, amino acid followed by serine or threonine, could use a very, very simple pattern that could facilitate very efficient transmission, including zoonotic transmission. And this part, so far, we haven't addressed fully in the influenza. So I just give you a broad idea, like an overview about the glycosylation variation by clade. Here, what I show here are 6 clades, basically the 6 families, gene set of subtypes in HIV family currently being selected in a vaccine design in HIV due to their clinical and epidemiological importance. So subtype A, B, C, D, and also their 2 recombinant family. First one, they call it CRF01, second one called CRF02. So these 2 are majorly actually impact the regional, it's not a global prevalence important. And the CRF01, actually, this is mostly on Asia, and actually CRF02, the mostly from West and West Central Africa. So from this kind of a comparison, which we select very, very representative sequences, actually, this is what we found. All the circulating actually clades of HIV, we found the glycosylation site, they have their lowest limit and the ceiling limit about the addition of the glycoprotein, which ranges actually 18 to 33, with a median value of 25. So this value, from first glance, you think, oh, okay, great, this is surveillance, this prevalence result. But actually, it's very, very helpful, helping us to select the candidate strain when we evaluate the vaccine efficacies. This is what we did in the past together with UNC researchers, together with experimentalists to see, okay, which strain globally we can use to represent the global diversity, that's including glycosylation site, in order to choose the appropriate test strain in vaccine efficacy evaluation. And there is one strain called HXB2. This is one of the most frequently used reference strain used in HIV field. And fortunately, the glycosylation site is about the median value. So when we selected the strain for the vaccine, for HIV vaccine evaluation, we always selected those strains with the glycosylation site around the median value, which is actually usually people don't think this at all. Another, this is just to give you overall, like by clade, and this is to give you more close look at the glycosylation site variation within one individual, because sometimes we need to not only care about the global prevalences, but also in order to improve so-called personalized medicine or precision medicine, we need to address individual differences. So just give you some close-out idea about the glycosylation site number changes and the frequency. So this shows over 4 years, actually there is a patient sampled 4 years, and each year we sampled about 10 sequences, which I only select, randomly select some sequences showing here as example. So here I only show about the envelope V1 loop, okay, and then the if there's a glycoprotein is found, the glycylation site found, I show it as N. And C, that is cysteine actually closing the V loop, is a V loop. So that actually, and the rest of the amino acid I just ignored, all the sequences actually I just ignored in order to emphasize the importance of the glycylation site and the length of the V loop that is actually due to the changes of the glycylation site. And then you see over the four time points over year, so we never see the same pattern of glycylation site in also actually the length of the variable loops. Okay, so this is a first like an individual example. Second example, just also repeat what I just described in other individual. So this is another individual also showing the V1 loop and the same strategy I show here. So you see all the kind of the length of the V loops with closing by C to C, cysteine to cysteine, and also the glycylation site, the precision location, they're so different. They're very different. They're never actually the same. Actually, this just across like a year. So this is brings out a lot of questions. So what should we do in order to address diversity and taking this into consideration into the vaccine design? And so far as you know, influenza, so glycoprotein is not that actually it's not heavily studied in this topic. But HIV is because it's one of the most glycosylated viruses in the world. So that's why we could start out with a glycylation analysis and experiments and test of a hypothesis, a lot of things starting from HIV. Then we can migrate the same system to the influenza. So another view about the glycylation site. Again, I choose all the current strains being selected for HIV vaccine evaluation, and also I use a group, a group O. So A, B, C, D, O, 1, O, 2, they're M group. I use O as out group. And then actually only focus on GP120, which is the region, most important region, immune recognizable region by the host immune system. So GP120, and then I use HXP2 again, there's a reference in the HIV, and then to track the pattern changes in terms of frequency of the glycylation site and also exact location across all the different the clades. So what do we see? Actually, there's two patterns actually that stand out. First one we called is a fixed pattern, which is I don't have any highlight. You see it is quite conserved between among all the clades. So they're suggesting actually there is a consistent immune pressure or evolution advantages preserving those sites. And also, actually, we found there's pattern which I highlight in the yellow here that shows as a shifting, we call it a shifting pattern of glycylation site. Basically, we see the glycylation site, the changes, actually very small variations in terms of frequency and also exact location. So those actually is very hard sometimes to see, but we develop a computer program that can automatically actually align those sequences like this. So for those shifting patterns, it looks from the first glance, we think, okay, it just maybe just run by random. But actually, if you actually compare with the reference sequences, the HXP2, especially if after we highlight the variable loop V1 to V5, which is very important for immune recognition, we found out actually those shifting patterns do not appear by chance. Okay, there are some reasons why those locations appear in this pattern. So when we compare the HXP2, especially we highlight the carbohydrate addition, which is biochemically very well defined, we find actually all the variable loops V1, V2, V3, V4, V5, they're mainly occupied by complex carbohydrate. And actually by the fixed pattern, which is don't have the highlight here, so mostly they're occupied by the high mannose type. So there's some importance why the viruses preserve this kind of fixed and the fixed and shifting pattern. So one reason because during the addition of carbohydrates, so it's due to different degree of exposure. So there's evolutionary advantages preserving those patterns. And also here I show there's three epitopes. So usually for the epitopes, people are more focusing on linear epitopes because it's easier to define. And for the spherical like epitopes, nonlinear epitopes is harder to find. However, here I just show you for the glycylation side, there are the three sites that contribute to the three epitopes actually that are part of the 2G12, which is actually the most frequently used broad crossing, the neutralizing antibody in HIV research. So for those three positions, actually you see that's occupied in the V3, V4 regions. So again, they're mainly actually highly they're mainly dependent on the minus type of the carbohydrate addition. And also this already later on experimentally proved there is some reason why those three positions, they rely on the mannose type carbohydrate addition because this could enhance the GP120 and the mannose interaction with the DC sign. So this is a kind of a pattern we see across different clades. The next question is actually we ask further anything actually related to the receptor usage differences. CCR5, CXCR4, and R5X4. This is three mainly used receptor in HIV entry. So we use here subtype ABCD because we don't have adequate sequences from the rest of the clades for this co-receptor usage. So all the co-receptor usage we use the data experimentally verified. So this is what we see actually. Most of the time, the pattern you see is very similar among the three population CCR5, CXCR4, and R5X4, which is actually a mix of these two co-receptor usage. Mostly you see is very similar between the three populations. But there are the two sides, actually they stand out. They're showing some differences. First one is at this position, we call it glycylation site 300 position. Actually, later on, they confirmed in experimental results based on our prediction, is that the 100%, actually 100% in this side in the CCR5, which is usually in the early stage of HIV infection. That's co-receptor usage. And this last degree appeared in a CXCR4, which is in a chronic HIV infection. And also, actually, it's a mixture between 64%, the same location, 64%. Actually, in a mixture, in a HIV population, they use actually mixtures R5 and X4, the population. This is one side, actually experimental verified. And the other side, we also see a similar trend, but we don't see any statistically significant yet. So this is a position, but it still is very interesting to look at the detail. So what's the advantages of actually having this site actually more highly expressed at this position, okay, this position. Another question we ask, okay, so just to show you across the clades what the differences of the sequence location and frequency, how this actually could help us to infer the epitopes locations. And next thing we are interested to know, actually, this is the first thing, we just did this for fun, but we found something very, very interesting. So we're still focusing on the V loops. I only show the V1 loop as example, but we did all the V loops. The result is very similar. So here, I choose HIV-1 and chimp, HIV-2, sooty monkey bee, and African green monkey. The reason why I choose this is because it's known for sure as actually the zoonotic transmission from chimp, the virus jumps from chimp to human, and then establishes the human transmission, and establish HIV-1. The similar thing, the virus from sooty monkey bee jumps to the human, establish HIV-2. So when I compare this kind of zoonotic transmission, it looks very interesting, the pattern. So first thing is actually a very outstanding observation here. We see when the sooty monkey bee jumped to the HIV-2, again, as I already showed, the glycylation site, actually the pink color glycylation site, and cysteine, the closing the V loop. So we see sooty monkey bee, and I compared with HIV-2, looks like HIV-2 has much more diversity and also the variation of glycylation site number and their locations. So this is less obvious when you compare the chimp to the HIV-1. It looks like from the first glance, it looks like the new host, because the changing environment from the natural host from monkey to human, okay, all of a sudden bring out a degree of diversity that we see in the glycylation site. So usually people don't see this actually as important, but we think is one of the zoonotic transmission barrier that the virus must cross in order to transmit to the new species. So the same thing actually we could think about influenza as well. So this is about the glycylation kind of a story we found so far, and we're still now actually investigating the same glycylation site pattern in a influenza. And also welcome if you have any collaborations in this regard as well. And just a brief conclusion is one thing we just see the result recently. It looks like for the glycylation site numbers in HIV, there is some subtypes, there is accumulation of glycylation site, but we don't see this in the influenza. It looks like influenza, there are some strategies very different from HIV, so that instead of accumulating, so in the influenza, we only see in H1N1, there's a little bit more obvious observation about accumulation glycylation site across all years. So next, mechanism I'm going to talk about, which we can also learn from HIV to infer better or improved the vaccine design for influenza, is about the recombination, which is similar to the reassortment in influenza. So why we care about the recombinant HIV? Because originally when HIV was discovered, nobody thought about this question before. That's why we never left the space in the nomenclature. Then, okay, we see a recombinant, how we can name it. It's just the scientists gradually cracked this mistake, over years, and find out, okay, recombination, it is a long time being ignored a problem. So before we start this recombination, just briefly, a review, this is a phylogenetic tree I just mentioned. As there's 9 genetic phylogenetic clusters. It looks very different from influenza phylogenetic cluster, which is the ladder shape in influenza. But HIV is more like the kind of star-like, and HIV actually is the diversity grow by time. So this inside the circle, this is what we see in 1990, and this is what we see about 2005. And actually the outside the circle actually is current HIV circulating strain. The diversity is, you see this is the tree, phylogenetic tree grow in time. And also the difficulty of current HIV vaccine design is that the recombinant actually blurs the subtype, the borders. I just to show you briefly, just using some examples, the recombinant will fill in the gaps between the clay. So that would fuzzy out that was actually actually blurs the border of different clades, so that so far the vaccine for each clade that may not work if you consider the same geographic region, but you have multiple sequences cross-circulate and that created recombination. So currently, very conservative estimate about HIV recombination is at least 20%. So that could be further divided into 50 clusters, which called families. Family we call the CRF circulating recombinant forms. In order to classify that, this is a very important nomenclature concept, which we currently challenging. Okay, so recombinant epidemic population, but they have to share identical mosaic structures within one CRF. This is current nomenclature for CRF, and it's the concept people using this concept in the vaccine design. Later on, I will mention why we challenge this concept. We think we should be modified to better reflect the changing dynamic picture of the HIV. So the question is that is recombination now in the past 3 years, the list of the family being identified actually is doubled. So our question is that is a situation getting too complex to be useful for the vaccine design or in the epidemiological surveillance? So we say actually our result is said it's not good enough. So how are we going to improve this? So I just give you an idea how I will work in this regard. First is about a CRF02, which is one of the most important recombinant families in HIV. So this is a very common in West and West Central Africa. So this is represented by the pink color here. So is our knowledge about CRF02 is evolving. In the past, 20 years ago, it said this is a classified as A, the gene subtype, and later on corrected, okay, we find something else inside, we don't know, so A plus U, and later on classified as G. However, this is not at the end of story, so this is so far, a lot of HIV groups stick with when they consider the vaccine design thing, okay, this is AG recombinant, that's all, so we use a subtype A and a subtype G in order to target the AG. But our finding found is actually is very, there's a lot of problems about this family alone. First thing, actually, we found most, when I say most, over 60% in this cluster, in this family, we found sequences, so-called CRF02 family members. They do not belong to this cluster based on the classical definition. Okay, second is that the CRF02 appears to be, has a very complicated history, okay, composing actually the recombination events between old circulating strains and its contemporary circulating strains. Why does matter? Because current vaccine design, whatever is, we focus on the circulating, the contemporary sequences, okay? But here, we do see some remnant fragment from this CRF02. They comes from they have very, very old origin, but we don't have the full-length genome because they die out from the circulation. So that's created a lot of problems, and also explains why CRF02 in most vaccine trials they fail. The first thing is actually the diversity we haven't addressed yet because of misgenotyping and also because by itself it contains some fragment. Currently we don't have the full-length actual genome in the vaccine design. So just to briefly let you know, the jumping profile hidden marker model, just one method, we draw this conclusion. So the chair mentioned to me, I'm almost there, so I have to speed up a little bit. But I welcome you to discuss with in detail about this method. We use this method mainly to lead to our findings. And this is the kind of a CRF02 family, but you see all of them actually we are listed here. They look very different. And this is our proposed prototype of the CRF02. This is the correct one. And this is just to show you part of this analysis result, actually showing, especially in part of the pole region. That is the part we found the old circulating subtype, which is haven't addressed in the current contemporary surveillance and vaccine design yet, and currently people just don't know. But this part, if you have vaccine targeting this part of the pole, for sure actually will fail because we don't have any references that could represent a global diversity in this region because it comes it has old origin and we don't have adequate samples. So a global view, I just mentioned there's many mistakes and we can correct. I just give an overview across genome and transmission pattern routes. So this is all the kind of also all the data has been normalized by the sequences number being identified in this in each transmission route. So we found actually in the GAG and the pole region on the envelope, actually there's a high degree of misgenotyping. That's lead to a lot of we call it information bias in your vaccine design because the pool we started from in the vaccine design is wrong. So this is just give you this idea we break into different groups. And further, across different geographic regions, actually if you see the darker color, that means the region has a higher rate of being misgenotyped. Also indication in this region, there is some unknown diversity we haven't identified so far, so we need to incorporate that better in the vaccine design. So what else we have missed? I just have one slide. Okay. What else we have missed? So this actually is very. This cartoon actually represents what we see in the current HIV the global epidemics, which is also we can use the same concept for the influenza. So this is what we see outside the circle. This is what we see in the contemporary HIV circulation, and inside this is what we see the old origin. So sometimes the strain, circulating strain, they're lucky. They have the oldest origin, but it reach out to the contemporary circulation. So, however, because the sampling bias for HIV mostly is more focusing on the Africa continent. So we probably didn't have those samples available. And some sequences actually, they're unfortunate. They're circulating for some time, but they die out in all the time, so that we cannot have any knowledge about this kind of die-out strains. But there are some older strains. We fortunately have their remnant presence that currently we don't have the full-length genome available, but they do have the fragmental presence in some recombinant genomes. For example, CRF01 and the one I just mentioned about CRF02, it has old origin of the A and old origin of the G. So this is part so far inside the circle. This is what we haven't incorporated in the current vaccine design. And we believe in the same strategy. Actually, we think we should improve the influenza or any kind of viruses vaccine as well because of sampling biases. So for the story and the lesson we learned from the HIV that we could use to infer the influenza vaccine design, first thing is make sure the pool we start with the vaccine selection is right. So unfortunately, currently we don't have a lot of good evaluation of circulating strain, their quality control in terms of sequence accuracy, and so this also leads to the sensitivity specificity test in your experiment, right? So second is we propose we should pay more attention about the glycylation protein, especially N-linked glycylation site, because this is what we see in the past in the HIV about the epitope. It could change the epitope, the exposure degree, and also could contribute to the zoonotic transmission, which is we haven't addressed yet fully in influenza. And also due to the high throughput sequencing, the next generation sequencing, we should incorporate the, for example, within patient diversity of HIV and also between population diversity. We should incorporate them at the to see the at the interface between host and the pathogen interface to see what we can find about, especially about the host factor, not only coding but also non-coding host factors that could regulate the immune recognition of viruses. So pathway, I will ignore this part. I just briefly show you this is what we found. This is a report of 2008 in Jackson Laboratory, and actually they found there is a non-structured protein NS1. Therefore, a C-terminal residue, the module pathogenicity. So we are able to narrow them by very simple modeling. We're able to down to one position just by doing through the modeling procedure, not necessary to do the point mutation. Not only validate, but also we could narrow it down into one position. So this is actually this is the lessons I believe there is a lot of space we can learn from HIV, which is one of the most variable pathogens in the world, 1 million times mutated faster than human genome. If we can learn about the mutation strategies and mechanism in HIV so that we can easily to combat with the influenza. For example, yesterday the two talks they mentioned about the fantastic work done by Todd Ross, actually currently in my university, University of Georgia. So he just moved there. Two talks mentioned about his the consensus design of the influenza strain. Actually, consensus strain for the HIV, we don't use this very often because of great diversity for the HIV. If you use a consensus for HIV, it will fail because it does not represent the natural strain of HIV. However, the strategy which we don't it does not work for HIV. It works for the influenza. So yesterday, two talks that actually shows the importance and efficacy of this the vaccine design, which is based on a consensus for the flu. So this is all. But last, just to show you the diversity of the influenza versus the HIV at different degree. Influenza HA. This is 1 year, 20 people. And this is global virus H3N2. This diversity showing all the sequences on one scale. And so this is global. And compared with HIV, which are only the similar genomic region, this is 9 sequences in one individual. Asymptomatic means here diversity of HIV is not expanded too much yet. And then this is in one city, Amsterdam, HIV-B, this is a 1 year. Okay, and then this is in one country alone. So compared with the global 96 about the same time, and actually they see 1 year global compared with one country. So that's actually what I have today. So this is all the people contribute to my work I just mentioned too. So if for any questions, I know I'll run this fast. Here's my contact info. Okay, thank you.
Are there any questions or comments? Comments, questions?
Yeah. Also welcome any discussion or potential collaborations how to migrate our system to more interesting viruses like influenza and HCV. Yeah.
We have 2 certifications for Ming Zhang for you here on the table.
Okay. Thank you. Oh, thank you.